Breathing is one of those things, isn't it? It's just fundamental, almost invisible. We do it constantly.
All the time.
Most of the time, you don't even give it a second thought. Air just, effortlessly moves in, effortlessly moves out. It feels inherently simple.
Seems that way.
But, like so many biological processes that seem simple on the surface, underneath there's this, absolutely incredible, complex, and deeply fascinating mechanism at work. So today we're doing a deep dive, really pulling back the curtain on exactly how that mechanism works.
That's right. What feels automatic, often, passive, is actually this highly coordinated system.
Right.
Muscle action, precise pressure regulation, the physical properties of our lungs, the chest wall, it's all working together.
Right.
Our goal in this deep dive, and we're using this really detailed expert source material you've got here.
Yeah, it looks pretty comprehensive.
It is. We want to move beyond just a surface level understanding. We're going to dissect the mechanics of pulmonary ventilation, that process of moving air into and out of the lungs with enough detail, enough rigor that you feel you're truly starting to master this.
Yeah.
Like someone who really works in this field.
Consider this source our roadmap then. It breaks it all down, piece by piece, figure by figure, even formula by formula.
Yeah.
Our mission is to navigate that map, pull out the vital insights, really understand the underlying pressures, the structures that make breathing possible, and equip you with that really detailed grasp. I'm genuinely excited to see just how intricate this seemingly simple thing is.
And I'm eager to guide that exploration. Because it's in those details. That's where you find the true marvel of the system. How these subtle, tiny changes in pressure can drive substantial airflow. How microscopic structures, even molecules, play absolutely critical roles. And how the system adapts, from just quiet breathing right up to maximum physical effort. Understanding these mechanics.
Yeah.
It's really the essential first step to appreciating the whole story of respiration.
Okay, let's unpack this then. Before we dive headfirst into the mechanics, the gears and levers, what's the whole system of respiration actually for at its core? What are its main jobs, according to our source?
Okay, fundamentally at the most basic level, respiration does two vital things. Get oxygen from the atmosphere to the body's tissues.
Right, the fuel.
Exactly. And then remove carbon dioxide from those tissues back to the atmosphere, the waste product. This constant exchange, it's essential for, cellular metabolism, for survival.
Makes sense.
But our source text here is careful. It breaks this whole process down into four major distinct components. They're interconnected, definitely, but distinct. It highlights that it's much more than just, breathing in and out.
4 components. Okay, tell me about them.
The first one, and this is our focus for this deep dive today, is pulmonary ventilation. Like we said, this is the mechanical process, the physical inflow of air, atmosphere into the lungs, specifically into the alveoli, those little air sacs, and then the outflow back out. It's the pump action.
So ventilation is the job of moving the bulk air, shifting the air mass. Got it.
Exactly. It's the delivery system. Get the air to the right place. The second component is the diffusion of oxygen and carbon dioxide.
The actual exchange.
Right. This happens right at the respiratory membrane.
Yeah.
That incredibly thin barrier between the air in the alveoli and the blood flowing through the pulmonary capillaries. This is where O2 moves into the blood, CO2 moves out.
Okay.
And our source clearly notes this is distinct from ventilation. It's governed by different principles, partial pressures, how easily gases diffuse. Stuff we'll cover later. It's not the mechanics we're focused on today.
So air moves to the alveoli. That's ventilation. Then the gases move across into the blood. That's diffusion.
Correct. Then component 3, transport of oxygen and carbon dioxide. Once those gases get into the blood, they need a ride, right? They need to be carried to all the tissues everywhere in the body, and CO2 needs to be carried back.
The circulation part.
Exactly. Handled by the circulatory system. Gas is dissolved in the plasma or bound to molecules like hemoglobin and red blood cells. Again, critical part of respiration, but separate from the mechanics of ventilation itself. Also for later exploration based on our source.
And the final component, number 4.
The fourth is the really complex process of regulation, the control system. Regulation of ventilation and other aspects of respiration. This involves nervous systems, chemical control systems in the brain, in the body, monitoring oxygen levels, CO2 levels, pH.
The body's thermostat for breathing.
Kind of, yeah.
Oh.
Adjusting the rate, the depth of breathing, even blood flow to perfectly match what the body needs metabolically. It's the conductor of the whole respiratory orchestra, a very detailed topic on its own, which our source handles separately.
Okay, so pulmonary ventilation, the mechanics we're digging into today. It's truly the essential first step. You can't have effective gas exchange, the diffusion, or the delivery, the transport, if the air isn't physically getting into the alveoli efficiently in the first place. It really is the foundation.
Precisely. Without that proper mechanical movement of air, the whole cascade just falls apart. Gas exchange, transport, even the signals that drive the regulation, it all gets disrupted. It is, as the source implies, the vital mechanical pump.
All right, that gives us the essential framework. We're zeroing in on that mechanical pump, pulmonary ventilation. Let's dig into how we actually expand and contract our lungs to move that air. Our source says there are two main ways the chest cavity volume changes.
Indeed. Think of the chest cavity like a bellows, right? And the lungs are inside. We increase and decrease the volume of that bellows in two main directions. Vertically, up and down, and anteroposteriorly, that's front to back.
The vertical movement, that sounds like the diaphragm's job.
It is. The diaphragm. That large, powerful, dome-shaped muscle right at the base of the chest cavity separates the chest from the abdomen. And it's the first, and for normal, quiet breathing, definitely the most important method.
How does it work?
During inspiration, when you breathe in, the diaphragm contracts. When it contracts, that dome shape flattens out and moves downwards, pushes down towards the abdomen.
Oh, okay.
This downward movement significantly increases the vertical dimension, the height of the chest cavity. It basically creates more space, top to bottom, for the lungs to expand into.
And for normal breathing out, is it actively pushing up?
For normal, quiet expiration, it's remarkably simple, it's almost entirely passive. The diaphragm just relaxes.
Really, no work involved.
Very little, typically.
Yeah.
See, the lungs themselves are elastic, like stretched rubber bands. The chest wall also has elasticity, and the abdominal contents that were pushed down, they naturally recoil upwards. So when the diaphragm relaxes, all these stretched elastic components just sort of snap back to their resting positions.
Like letting go of a stretched spring.
Exactly. This recoiling action compresses the lungs from below and around, pushing the air out. No active muscle contraction is usually needed for just quiet, everyday breathing out.
Huh. Fascinating that half the normal cycle is just passive recoil. Okay, so diaphragm handles vertical expansion, and passive recoil handles normal vertical contraction. What about the second method you mentioned, changing the front to back diameter?
Right, that involves moving the rib cage. If you look at figure 38:1 in the source, you can see the ribs. They naturally angle downwards from back to front when you're at rest.
Yeah, I see that, sloping down.
This means the sternum, the breastbone at the front, sits relatively close to the spine at the back, limits the front to back depth. Now, when the rib cage is elevated,
Lift it up.
yes. The ribs lift and they also rotate outwards slightly. Because they're angled downwards and forwards to begin with, this upward and outward movement makes them project more directly forward. Think of it like lifting bucket handles.
Okay, yeah, pivoting up and out.
Right. And this pushes the sternum away from the spine. This action significantly increases that anteroposterior diameter, the front to back depth of the chest cavity.
And does the source quantify how much it increases?
It does. It notes this front to back diameter can increase by about 20% during a maximum inspiration compared to a maximum expiration, all due to that rib cage elevation. And then depressing the ribs, pulling them back downwards and inwards, reverses this, decreases the front to back diameter, and helps with expiration.
So lifting the rib cage helps you breathe in, pulling it down helps you breathe out. Makes sense.
Precisely. And this brings us to the specific muscles involved. Now, we said the diaphragm is the main player for quiet inspiration, and quiet expiration is passive. But during heavy breathing, forced breathing, like when you're exercising really hard or coughing, that passive elastic recoil just isn't fast enough or strong enough.
Right, you need to move more air faster, so you need active muscle work for forced expiration too.
Absolutely. For a forced expiration, the main muscles doing the work are the abdominal muscles, particularly the abdominal recti or abs. When they contract forcefully, they squeeze the abdomen, pushing the abdominal contents upwards against the underside of the diaphragm.
Ah, pushing the diaphragm up from below?
Exactly. Pushes it higher into the chest than its normal relaxed position, powerfully compresses the lungs, plus the internal intercostal muscles also kick in. They lie between the ribs, but they angle in the opposite direction to the external intercostals.
Okay.
When the internal intercostals contract, they pull the rib cage downwards and inwards, further shrinking the chest volume. Powerful expiration.
Okay, forced expiration. Abdominals pushing the diaphragm up, internal intercostals pulling ribs down. Got it. What about forced inspiration then? What muscles help lift the rib cage more forcefully than normal, beyond what the diaphragm does?
Well, essentially, any muscle that can elevate the rib cage is classed as an inspiratory muscle when we talk about forced breathing. The most important ones for lifting the ribs are the external intercostal muscles.
Okay, the ones between the ribs angling the other way.
That's them. Figure 38:1 shows they angle downwards and forwards from the rib above to the rib below. When they contract, they pull the upper rib forward and upward relative to the lower one. Or you can think of them pulling both ribs towards each other. This creates an upward and outward leverage on the whole rib cage, lifting it. Significantly increases chest volume for a deep, forceful breath in.
So external intercostals pull up and out, internal intercostals pull down and in. The opposite angles make sense for their opposite actions.
Exactly right. And for a truly maximal inspiratory effort, like gasping for air, our source mentions several helper or accessory muscles chip in. These include the sternocleidomastoid muscles in your neck. They can lift the sternum upwards. The anterior serrati muscles on the sides. They can lift many of the ribs. And the scalene muscles, also in the neck, they lift the first two ribs specifically. All contribute to that maximum possible chest expansion.
What's really interesting here is how the body layers this muscle recruitment. Quiet breathing is incredibly efficient, mostly just the diaphragm and then passive recoil. Costs very little energy.
Right.
But when the demand goes up, it activates these additional specialized muscle groups, the external intercostals and those accessory muscles for deeper breaths in, and the abdominals and internal intercostals for forceful breaths out. It's a clear step-up mechanism, going from mostly passive expiration to actively, forcefully changing chest volume in both directions. This really highlights the system's, capacity and reserve power.
It really does. It shows how ventilation can be scaled up dramatically, meeting demands far beyond resting levels, all through this coordinated muscular action.
Okay, so muscles change the volume of the chest cavity. We got that. But how does changing the volume actually pull air into lungs or push it out? Air movement has to be driven by pressure differences, right?
Absolutely, 100%. Just like water flows downhill, air moves from a region of higher pressure to a region of lower pressure. The entire mechanics of ventilation depend on creating and managing specific pressure gradients within the respiratory system. And our source details the key pressures we really need to understand to get this. There's the pleural pressure, the alveolar pressure, and then the critical difference between them, the transpulmonary pressure.
All right, let's tackle them one by one. Pleural pressure first. Where exactly is that pressure and why is it so important?
Okay. The lungs themselves, remember, are elastic. They naturally want to recoil inwards, collapse down like a stretched balloon wanting to deflate. Now, they aren't directly attached to the inside of the chest wall, except right at the hilum, where the airways and blood vessels go in. Instead, they're sitting inside the chest cavity. And between the outer surface of the lung, that's covered by a membrane called the visceral pleura, and the inner surface of the chest wall, covered by the parietal pleura, there's this incredibly thin potential space. That's the pleural space, and it contains just a tiny amount of pleural fluid.
What's the fluid for? Lubrication.
Lubrication, yes. It allows the lung surface to slide really smoothly against the chest wall as we breathe. But critically, this fluid is constantly being sort of sucked away or pumped out by lymphatic vessels in that pleural space. This constant removal maintains a slight negative pressure or suction within the pleural space relative to the atmospheric pressure outside.
Ah, so it's like a vacuum seal almost.
That's a good way to think about it. This negative pressure effectively glues the outer surface of the lungs to the inner surface of the chest wall. So when the chest wall expands or contracts, the lungs are pulled along with it. They have to follow.
It's not a physical attachment, but a pressure-based tether holding them together. And the pressure in this pleural space is the pleural pressure.
Exactly. Pleural pressure is the pressure measured in that thin pleural space. And normally, it's negative. Our source, like many physiology texts, uses centimeters of water, centimeters H2O, to measure these fairly small pressures. And it uses atmospheric pressure as the zero reference point. So negative means below atmospheric.
How does this negative pressure change when we breathe?
Okay, look at figure 38:2 from the source material. Shows this clearly. At the very beginning of a normal, quiet inspiration, just before air starts to flow in, the pleural pressure is already negative, typically around -5 centimeters H2O. This is the resting section, the baseline negative pressure that holds the lungs open to their normal resting volume, what we call the functional residual capacity.
Okay, -5 at rest.
Then, as you inspire, the chest cage expands, pulled outwards by the diaphragm contracting downwards, and maybe the external intercostals lifting the ribs slightly. This expansion pulls outwards on that parietal pleura lining the chest wall. Since the lungs are glued to the chest wall by that negative pressure,
They get pulled outwards, too.
right, they get stretched open. This outward pull increases the volume of that tiny pleural space slightly, which increases the suction. It makes the pleural pressure even more negative. During a normal inspiration, it typically drops down to about -7.5 centimeter H2O by the end of the breath in.
So inspiration. Pleural pressure goes from -5 down to -7.5, more negative.
Exactly. And then during normal passive expiration, the chest wall and the lungs recoil back inwards. This reduces that outward pull on the pleura, the suction decreases, and the pleural pressure returns to its resting, less negative value of about -5 centimeter H2O.
Okay, that's the pressure outside the lungs in that pleural space, keeping them stuck to the chest wall and pulling them open. What about the pressure inside the actual air sacs, inside the alveoli, the alveolar pressure?
Right, that's the alveolar pressure. The air pressure right inside the lung alveoli. Now, when your glottis, the opening at the top of your windpipe, is open, and there's no air flowing either in or out, like that brief pause between breaths, or if you just hold your breath with your airway, open the pressure throughout the entire respiratory tree all the way down to the alveoli, becomes equal to the pressure outside your body, equal to atmospheric pressure.
So we call that zero.
We use that as our reference point, yes. 0 centimeter H2O.
So if you're just sitting here breathing normally, alveolar pressure briefly hits zero right between each breath.
Makes sense.
Now, to get air to flow into the lungs during inspiration, the pressure inside the alveoli must become slightly lower than the pressure outside, atmospheric
You
pressure.
got to create a gradient. Exactly. As the chest cavity expands and pulls the lungs open, the volume inside the alveoli starts to increase. Boyle's law tells us if the volume goes up, the pressure must come down, assuming constant temperature. Figure 38-2 shows that during a normal inspiration, the alveolar pressure drops just slightly to about -1 centimeter H2O, just 1 centimeter of water below atmospheric.
That's it. That tiny pressure drop pulls air in.
That tiny negative pressure gradient is enough. It's enough to pull about half a liter, 500 milliliters of air into the lungs over the roughly 2 seconds of a normal inspiration.
Wow. Okay, and expiration.
Opposite logic. To push air out of the lungs during normal expiration, the alveolar pressure has to rise slightly above atmospheric pressure. As the chest and lungs recoil inwards, the volume of the alveoli decreases. This compresses the air inside, causing the pressure to increase. Figure 38-2 shows it rises to about +1 centimeter H2O, just 1 centimeter of water above atmospheric. This slight positive pressure gradient is what forces that half liter of air back out over the 2 to 3 seconds of normal expiration.
Okay, this is clarifying. Pleural pressure is always negative. Gets more negative during inspiration. Alveolar pressure swings slightly negative during inspiration, like -1, to pull air in, and slightly positive during expiration, like +1, to push air out, hitting zero between breaths. And the relationship between these two pressures, that must be key.
Absolutely critical. The difference between the alveolar pressure inside the lung and the pleural pressure outside the lung in that space is defined as the transpulmonary pressure. Our source describes this as essentially the measure of the elastic forces in the lungs that are trying to make the lungs collapse at any given moment. It's the recoil pressure. Think of it as the pressure difference across the wall of the lung itself.
Can you give me an example using those numbers from figure 38-2, like at the start of a breath?
Sure. At the very beginning of inspiration, alveolar pressure is 0 centimeter H2O, and pleural pressure is, again, 5 centimeter H2O. The transpulmonary pressure is alveolar minus pleural, so 0 minus 9 to 5 equals plus 5 centimeters H2O. This positive 5 centimeter H2O is the pressure difference across the lung wall that's holding the lungs open against their natural elastic recoil at their resting volume, that FRC we mentioned.
A +5 pressure holding them open at rest, and at the peak of a normal inspiration.
Okay. During inspiration, alveolar pressure drops to -1 centimeter H2O, and pleural pressure becomes more negative, down to about -7.5 centimeter H2O. So the transpulmonary pressure is now -1 minus -7.5. That equals plus 6.5 centimeter H2O.
Ah, it increased from +5 to +6.5.
Exactly. It's this increase in the transpulmonary pressure from +5 to +6.5 that represents the increased stretching force across the lung wall. It's what overcomes the lung's increasing elastic recoil as it expands and pulls the lungs further open during inspiration.
Okay, here's where it gets really interesting, I think. It's the subtle dynamic interplay. The always negative pleural pressure acts like that external suction cup, keeping the lungs tethered to the expanding and contracting chest wall, preventing total collapse. The tiny swings in alveolar pressure, just below or just above atmospheric, are the direct result of those chest mechanics, creating the immediate gradient that drives air in or out. And then the transpulmonary pressure, that difference between inside and outside the lung wall, is like the net inflating pressure that's constantly battling the lung's own desire to shrink back down, its elasticity, this subtle dance of pressures. That's the real engine of ventilation, isn't it?
It truly is. A brilliant piece of biomechanical engineering. And that whole concept of elastic recoil and stretching brings us right to the next point.
Yeah.
How much do the lungs actually stretch when that transpulmonary pressure changes?
That sounds exactly like the definition of compliance.
It is. Pulmonary compliance is precisely that. It's a measure of the lung's distensibility, how easily it stretches. It's formally defined as the change in lung volume for each unit increase in transpulmonary pressure.
How much bang for your buck, pressure-wise?
Kind of, yeah.
Yeah.
And a key point from the source is that this is measured after giving the lung enough time, like 10 to 20 seconds, to reach its full expansion at a new steady pressure. You have to let it reach equilibrium.
What's a typical value for lung compliance in a healthy adult?
The total compliance of both lungs together averages about 200 milliliters of air per centimeter of water transpulmonary pressure. So 200 millisimidia H2O.
What does that mean practically?
It means if you increase that transpulmonary pressure by just 1 centimeter H2O, a very small pressure change, the lung volume will expand by 200 milliliters once it settles. Think about that. Only 1 centimeter of water pressure difference across the lung wall expands its volume by nearly a cup full of air. That really gives you a feel for how stretchy, how distensible the lungs normally are.
Yeah, that is surprisingly flexible. And this relationship, volume versus pressure, it's shown in a compliance diagram.
Yes. Figure 38-3 shows the compliance diagram for the lungs. It plots lung volume on the vertical axis against the pleural pressure on the horizontal axis. Remembering pleural pressure changes directly reflect transpulmonary pressure changes. And you see right away, it's not just a single straight line. There's one curve for inspiration as volume increases, and a distinctly different curve for expiration as volume decreases.
And that gap between the curves, that's called hysteresis.
Correct. That separation between the inspiratory and expiratory limbs is hysteresis. It shows us that the pressure-volume relationship isn't perfectly linear. It's not like a simple spring. It depends on whether the lung is currently expanding or contracting. Understanding why those curves are separate tells us something important about the energy involved. Some energy is lost during the cycle, partly due to tissue friction, but mostly due to the effects of surface tension at the air-fluid interface changing as the lung inflates and deflates.
How is that diagram usually recorded?
Typically, they change the pleural pressure in small, discrete steps, hold it steady, wait for the lung volume to stabilize at that pressure, take the measurement, and then move to the next pressure step. That's how you trace out those curves.
Okay, so compliance tells us how much the lungs stretch for a given pressure change. And the diagram with its hysteresis shows this relationship is complex, not perfectly elastic. What physical properties within the lung tissue actually give it these specific elastic characteristics?
Right. The elastic behavior which determines compliance comes from two main types of elastic forces within the lung structure itself. The first type is the elastic forces of the lung tissue.
What's that made of? Like fibers?
Exactly. It's determined mainly by this intricate, interwoven network of elastin and collagen fibers that make up the lung's structural framework, the parenchyma. When the lungs are deflated, these fibers are sort of bunched up, kinked, in an elastically contracted state. As the lungs expand during inspiration, these fibers get stretched out, unkinked. And just like stretching a rubber band, they exert an increasing elastic force resisting further expansion.
So the tissue itself acts like a stretchy, expanding scaffold.
That's a good way to put it. Now, the second type of elastic force is actually much more complex. And maybe surprisingly, it's the more significant of the two. These are the elastic forces caused by surface tension.
Surface tension from fluid inside the lungs?
Exactly. Surface tension of the thin layer of fluid that lines the inner surfaces of all the alveoli and the other tiny air spaces.
How do we know that surface tension force is actually more important than the tissue's own elasticity?
Our source provides a really elegant and powerful demonstration of this in figure 38-4. It compares the compliance diagram of lungs filled with saline solution, basically salt water, versus lungs filled with normal air.
Saline versus air. Okay, what does that comparison show?
Well, when the lungs are filled with saline, there's no air-fluid interface inside the alveoli. It's fluid-filled. So that eliminates any elastic forces caused by surface tension. You're only measuring the elastic forces of the lung tissue itself.
Makes sense.
In contrast, when the lungs are filled with air, you have that critical air-fluid interface. So the surface tension effect is fully active, in addition to the tissue forces.
And the difference in the pressure needed to inflate them is big.
Strikingly big. The transpleural pressures needed to expand the air-filled lungs to any given volume are about 3 times as great as the pressures needed to expand the same lungs filled with saline solution.
Wow, 3 times!
That comparison is a fundamental insight from our source. It tells us that the tissue elastic forces alone, measured in the saline lung, account for only about 1/3 of the total lung elasticity in normal air-filled lungs. The other 2/3 of the elastic force trying to collapse a lung comes from that fluid-air surface tension in the alveoli.
Okay, that's a critical point. Goes against intuition, maybe? You'd think the tissue structure provides most of the springiness, but no, it's the surface tension of that thin fluid lining that's the dominant force resisting lung expansion and making the lungs want to collapse. That's a major takeaway from the source for really understanding compliance.
It is indeed a key insight. And it highlights just how incredibly important that fluid lining is. And, even more critically, what controls the properties of that fluid lining. Which brings us right to a critical substance.
What does control that dominant surface tension force? And you hinted earlier, why does it increase massively if something called surfactant isn't there?
Yes, surfactant. This substance is absolutely indispensable for normal, easy breathing. But first, let's just quickly refine our understanding of that surface tension principle, specifically in these tiny alveoli.
Okay, lay it out. Surface tension in the alveoli.
So water molecules, they really like each other. Strong mutual attraction. At a surface where water meets air, this attraction pulls the surface molecules together, trying to make the surface area as small as possible. That's surface tension. It creates a contractile force. Now imagine that thin film of fluid lining the inside walls of every single alveolus. The water molecules in that film are pulling on each other. This pulling generates a force that tends to push air out of the alveolus, making the alveolus want to shrink, to collapse. This is the surface tension elastic force of the lungs we just talked about.
Okay. And surfactant is the antidote?
Precisely. Surfactant is a substance that acts as a surface active agent, meaning it dramatically reduces the surface tension of water. It's made and secreted by special cells right there in the alveolar walls, called type 2 alveolar epithelial cells. They're granular-looking cells, make up maybe 10% of the alveolar surface area, and they produce these lipid-rich packages containing the surfactant.
What exactly is surfactant made of, chemically?
It's a complex mix, actually. Contains phospholipids, proteins, ions. But the single most crucial component, the real workhorse for reducing surface tension, is a specific phospholipid called the palmitoyl phosphatidylcholine, DPPC for short.
And how does DPPC actually cut down the surface tension?
It's down to its molecular structure. It has parts that like water, hydrophilic, and parts that hate water, hydrophobic. So it doesn't just dissolve uniformly in that fluid layer. Instead, part of the molecule dissolves, but the hydrophobic part spreads out right on top of the fluid surface at the interface with the air. This layer of surfactant molecules gets in between the water molecules, disrupting their strong attraction to each other, and that's what lowers the surface tension.
Can you give us a sense of the numbers? How much reduction are we talking about?
The source gives specific values, which are quite dramatic. Pure water has a surface tension around 72 dynes per centimeter. The fluid lining the alveoli, if it lacks surfactant, would be maybe around 50 dynes per milliliter. But with a normal amount of surfactant present, the surface tension in the alveolar fluid is slashed way down to a range of between 5 and 30 dynes per milliliter. It varies a bit because the surfactant molecules get more concentrated as the alveolus shrinks during exhalation, making it even more effective at smaller sizes. But think about that drop from 50 down to potentially 5.
That huge reduction must drastically change the pressure inside the alveoli that's trying to make them collapse.
It absolutely does. Remember, surface tension trying to collapse an alveolus generates a positive pressure inside it. There's a physical principle based on the law of Laplace that our source applies here. The pressure trying to collapse an alveolus is equal to 2 times the surface tension divided by the radius of the alveolus.
Pressure 2 tension radius. Okay.
Let's plug in some numbers. For an average-sized alveolus, maybe 100 micrometers radius, lined with normal surfactant, let's use an average surface tension of, say, 20 dynes per milliliter for this example.
Right.
The pressure trying to collapse it calculates out to about 4 centimeter H2O. That's the intrinsic collapsing tendency with surfactant.
Okay, 4 centimeter H2O collapsing pressure normally. What if there's no surfactant? Like if it was just plain fluid around 50 dynes per meter?
If those same alveoli were lined with fluid at 50 dynes per meter surface tension, The calculated collapsing pressure jumps up to about 18 centimeters H2O. That's 4.5 times greater.
Whoa. So surfactant directly slashes the collapsing pressure, making it far, far easier for our breathing muscles to inflate the lungs against that force.
Exactly. Without surfactant, the sheer effort, the work required just to overcome surface tension and pop open the alveoli with each breath would be immense. Probably unsustainable.
And that formula, pressure to tension radius, it points to another really crucial factor. The size of the alveolus matters a lot.
It does. The formula clearly shows pressure is inversely related to the radius. This is another key insight from the source material. The smaller the alveolus, the greater the pressure trying to collapse it due to surface tension.
Yeah.
This effect is actually amplified as alveoli naturally shrink a bit during exhalation.
Smaller size equals a bigger collapsing problem, and this is where we connect to premature babies, right?
Precisely. Small premature infants often have significantly smaller alveoli compared to full-term babies, sometimes less than a quarter of the adult radius. And critically, the production and secretion of surfactant by those type 2 cells doesn't usually kick in properly until quite late in pregnancy, typically between the 6th and 7th months, sometimes even later.
So a baby born very prematurely could have tiny alveoli and little or no surfactant. A double whammy.
That's often exactly the situation. A severe surfactant deficiency combined with smaller alveoli. The result. An extreme tendency for their lungs to collapse. The collapsing pressures can be 6 to 8 times greater than in a normal adult. This condition is known as respiratory distress syndrome of the newborn, or RDS.
That's very serious.
Extremely. Our source emphasizes that without immediate aggressive medical intervention, often including continuous positive pressure breathing, CPAP, to physically splint the airways and alveoli open, and sometimes administering artificial surfactant directly into the lungs, RDS is frequently fatal.
Wow. Okay, this makes it absolutely crystal clear. Surfactant isn't just a nice bonus. It's essential for life, especially for keeping those smallest lung units, the alveoli, open against the huge forces of surface tension, particularly when they are also small. That single substance, its presence or absence, explains a major life-threatening respiratory emergency in newborns. That's a vital takeaway from the source if you want to master this material.
It's a truly powerful illustration. A specific molecule being fundamental to the entire mechanical process of breathing.
Okay. We've gone deep on the lungs' own elasticity, the battle between collapsing forces like surface tension and the inflating pressures. But, the lungs don't exist in a vacuum. They're inside the thoracic cage, the chest wall, which isn't perfectly floppy either. How do the mechanical properties of the chest cage itself affect the whole system?
That's a crucial consideration, absolutely. The thoracic cage isn't just a passive box. It has its own elastic properties, its own stiffness, and even some viscous resistance to movement. Expanding the chest cavity itself requires muscular effort, even if you imagine the lungs weren't inside. Think about bending those ribs versus just stretching the soft lung tissue.
So that compliance measurement we talked about earlier, the 200 millisimeter H2O, was that just for the lungs alone?
Yes, that value is specifically for the lungs when isolated, removed from the chest. When we talk about the compliance of the entire pulmonary system, that is, the lungs and the thoracic cage working together, the picture changes quite a bit. This combined compliance is typically measured in a person whose breathing muscles are totally relaxed, or even paralyzed, by carefully forcing air into the system and recording the pressures and volumes.
And how does the compliance of the combined system compare to the lungs alone?
Well, to inflate the combined lung-thorax system, our source tells us it requires almost twice as much pressure compared to inflating just the lungs alone after they've been removed from the chest.
Twice the pressure needed.
Almost twice. And as a direct result, the compliance of the combined system is about half that of the lungs alone. It averages around 110 millisimeter H2O for the combined system versus the 200 millisimeter H2O we saw for the lungs by themselves.
So adding the chest cage makes the overall system roughly twice as stiff or half as compliant as the lungs are by themselves.
Effectively, yes. The chest cage adds significant resistance to the system's expansion. And the source also highlights something important. The chest cage imposes limits that become especially noticeable at very high lung volumes, near total inflation, or at very low lung volumes, near complete deflation. At these extremes, the compliance of the combined system can plummet, sometimes dropping to less than 20% of the compliance of the lungs alone. The chest wall basically acts as a much more rigid boundary at the edges of our breathing capacity.
This leads us naturally, I think, to the concept of the work of breathing, the actual energy the body has to spend to do all this mechanical ventilation.
Exactly. Breathing isn't metabolically free. It costs energy. Now, in normal, quiet breathing, as we established, most of the active muscle work happens during inspiration, primarily the diaphragm contracting. Expiration is largely passive, driven by the elastic recoil of both the lungs and the chest cage snapping back to their resting positions.
So the main work is done just to expand the system against those elastic forces. What exactly is that energy spent on during inspiration? Can we break it down?
Yes, the energy used during inspiration goes towards overcoming several different types of resistance. Our source divides the work of inspiration into three main components or fractions. First, and this is the largest part, is compliance work, which is also called elastic work. This is the energy needed to expand both the lungs and the chest wall against their combined elastic forces, basically overcoming the stiffness of the whole system. This accounts for about 65% of the total work of inspiration.
Okay, 2/3 just stretching the springs. Makes sense. What's the second component?
Second is tissue resistance work. This is the energy required to overcome the viscosity, or you could think of it as the internal friction, of the lung and chest wall structures themselves as they move and slide against each other during breathing.
Like the tissues rubbing.
That's a simplification, but it captures the idea of frictional resistance to the tissues moving. This is a relatively smaller fraction, accounting for only about 5% to 10% of the work.
Okay. And the third fraction?
The third is airway resistance work. This is the energy needed to overcome the resistance to the flow of air as it moves through all those respiratory passages. The trachea, bronchi, bronchioles, down into the lungs. This makes up about 25% to 30% of the total work.
Okay, let me see if I got that. Roughly 2/3 stretching the elastic lungs and chest, maybe 5–10% overcoming tissue friction, and about 1/4 pushing the air through the tubes. That actually sounds pretty efficient overall.
It is remarkably efficient under normal circumstances. Our source states that during normal quiet respiration, the whole process of ventilation uses only about 3% to 5% of the total energy expended by the body. It's a very low metabolic cost for a function that's literally keeping us alive moment by moment.
I assume that cost changes dramatically under stress.
Absolutely. During heavy exercise, when your minute ventilation might increase 20-fold or more, the amount of energy required just for breathing can increase massively, up to 50-fold. This is the really crucial point for understanding limits. Respiration goes from being a negligible energy drain to potentially a very significant one under high demand.
Wow, 50 times more energy just to breathe.
And this increase in energy cost is far, far greater if a person has any kind of lung disease that either increases airway resistance, like asthma or COPD.
Right, harder to push air through the tubes.
Or decreases pulmonary compliance, like pulmonary fibrosis, where the lungs become stiff and hard to stretch.
So respiratory disease doesn't just make breathing feel harder, it makes it objectively much more energy intensive for the body.
Precisely. Our source points out that the energy cost of breathing itself can become a major limiting factor on the intensity of exercise a person can perform. If your respiratory muscles are consuming a huge chunk of your available energy just trying to move air in and out, there's far less energy left over for your leg muscles or arm muscles to do the actual physical work.
That's a key insight. It highlights how conditions we might think of as just airway problems, like asthma, can profoundly impact overall physical capacity, partly because they dramatically increase this hidden workload of breathing.
It really connects the mechanics to the metabolism. If we link this back to the bigger picture, breathing feels effortless, and normally it costs almost nothing energy-wise. But it's this incredibly efficient system with a huge reserve capacity that, under stress or in disease, reveals itself as potentially a major energy consumer. Understanding this energy cost is vital to appreciating the real impact of respiratory conditions and the body's ultimate physical limits. This is another one of those points from the source that's key for a deeper professional level, understanding how the hidden metabolic burden of breathing can actually become the primary limitation in disease or extreme effort.
It absolutely shifts your perspective from just moving air to managing a dynamic energy budget. Okay, so we've explored the muscles, the pressures, the elasticity, the surface tension, the energy cost. But how do we actually quantify all this? How much air is moving? How much capacity someone has? How do doctors or researchers get specific measurements?
Right. The standard technique they use is called spirometry. It's really the basic, fundamental tool for studying pulmonary ventilation. It worked by recording the volumes of air that move into and out of the lungs during different breathing maneuvers.
Spirometry. Okay. How does a typical spirometer actually work?
Figure 38 of 5 in the source shows a classic type. Essentially, you have a drum inverted over a tank of water with a counterbalancing weight. Inside the drum is a breathing gas, usually air or oxygen. The person breathes through a tube connected to this gas chamber.
Right.
As they inhale, they draw gas out of the drum, so the drum falls. As they exhale, they push gas into the drum, and the drum rises. This up and down movement is recorded either on a rotating paper chart or digitally. The recording directly tracks the changes in the volume of gas inside the drum, which perfectly mirrors the volume of air entering or leaving the person's lungs.
And that recording gives us a picture, a graph of different lung volumes over time.
Exactly. That recording is called a spirogram. Figure 38-6 shows a typical spirogram, illustrating various breathing efforts and the volumes associated with them. Now, for ease of description and analysis, the air volumes moved during breathing are traditionally divided into 4 primary volumes. And then various combinations of these volumes are called pulmonary capacities.
Volumes and capacities, got it.
And it's important the specific values shown in figures 38-6 and table 38-1. Those are average values for a young adult man. As we'll discuss, these values vary considerably depending on the individual.
Okay, let's break down those 4 fundamental volumes first. What are they?
All right, the 4 main pulmonary volumes are
Tidal volume, VT. This is the volume of air you inspire or expire with each normal, quiet breath, just regular breathing. In an average healthy adult man, this is around 500 millimole, half a liter.
Okay, VT 500 milliliters normally.
Number 2, inspiratory reserve volume, IRV. This is the extra volume of air you can possibly inhale above your normal tidal volume if you make a maximum effort. So take a normal breath in, then inhale as much more as you possibly can. That extra amount is the IRV. It averages about 3,000 milliliters. Quite large.
3 liters reserve on the way in.
Okay. Number 3, expiratory reserve volume, ERV. This is the flip side. It's the maximum extra volume of air you can forcefully exhale after letting out a normal tidal breath. So breathe out normally, then push out as much more air as you possibly can. That extra amount is the ERV. It's typically smaller than the IRV, around 1,100 milliliters in men.
About a liter reserve on the way out.
Right. And number 4, residual volume, RV. This is the volume of air that remains trapped in your lungs even after you've performed the most forceful expiration possible. You simply cannot exhale this air. It averages around 1,200 milliliters.
So even when you breathe out absolutely everything you can, there's still over a liter of air left inside. That residual volume seems pretty important.
It is. It's crucial for keeping the smallest airways and alveoli from collapsing completely at the end of expiration. It also helps buffer changes in blood gas levels between breaths. But as the source clearly notes, precisely because you cannot expire this air, the residual volume cannot be measured directly using simple spirometry. That's a key limitation.
Okay, those are the 4 volumes. Tidal, inspiratory reserve, expiratory reserve, and residual. What about the capacities? You said capacities are combinations of these volumes.
Exactly. Pulmonary capacities are just sums of 2 or more volumes, and they're often useful for describing larger functional ranges of breathing. There are 4 main ones.
Inspiratory capacity. I see. This is simply the tidal volume plus the inspiratory reserve volume, VT plus IRV. It represents the maximum amount of air you can breathe in, starting from the end of a normal, quiet exhalation. For our average man, it's about 500 plus 3,000 for 3,500 milliliter.
Total amount you can inhale from rest.
Got it. Second, functional residual capacity, FRC. This equals the expiratory reserve volume plus the residual volume, ERV plus RV. This is the amount of air that remains in the lungs at the end of a normal quiet expiration. It's your resting lung volume. Averages about 1,100 plus 1,200 equals 2,300 milliliters. The source highlights this FRC as particularly crucial physiologically. It's the reservoir air in the lungs that ensures gas exchange can continue relatively smoothly even between breaths, preventing wild swings in blood oxygen and CO2 levels.
The buffer volume.
Third, vital capacity, VC. This is the sum of the inspiratory reserve volume, the tidal volume, and the expiratory reserve volume, IRV plus VT plus ERV. It represents the maximum amount of air you can possibly move in and out of your lungs voluntarily. You measure it by taking the deepest possible breath in and then exhaling as forcefully and completely as possible. That total exhale volume is the VC. It's about 3,000 plus 500 plus 1,100 plus 600 milliliters. Notice also VC is the same as inspiratory capacity plus expiratory reserve volume, IC plus ERV. 3,500 plus 1,100 is 4,600.
The biggest breath you can possibly take and expel.
That's it. And finally, fourth, total lung capacity, TLC. This is the absolute maximum volume to which your lungs can be expanded with the greatest possible inspiratory effort. It's all the air the lungs can hold. It equals the vital capacity plus the residual volume, VC plus RV. For our average man, that's about 4,600 plus 1,200 plus 5,800 milliliters. You can also calculate it as the functional residual capacity plus the inspiratory capacity, FRC plus IC. 2,300 plus 3,500 equals 5,800. It all adds up.
Okay, that structure makes sense. 4 core volumes combined in different ways to make 4 functional capacities. And you mentioned these average values vary quite a bit between people.
Yes, definitely. As Table 38.1 in the source shows, these values are typically 20% to 30% lower in women compared to men, mainly due to body size differences. They also vary significantly with a person's height and build, their level of physical fitness, their age. Lung volumes generally tend to decrease somewhat with aging, and even things like altitude acclimatization. So these numbers are useful averages, but interpreting them clinically always requires comparison to predicted values based on the individual's characteristics.
The source also introduces standard abbreviations for all these, right? Like BT, FRC.
Right, Table 38.2 lists the common abbreviations used in pulmonary function testing.
Mm-hmm.
BT, IRV, ERV, RV for the volumes, and IC, FRC, VC, TLC for the capacities. These are the standard language used in formulas and clinical reports. And those algebraic relationships we just discussed can be written concisely using these symbols like VC-IRV plus VT plus ERV or FRC-ERV plus RV.
Okay. Now you pointed out something important. Residual volume, RV, can't be measured by simple spirometry because you can't breathe it out. And since functional residual capacity, FRC, includes RV, FRC-ERV plus RV, that means FRC also can't be measured just by breathing into a standard spirometer.
Correct.
So how do they determine those crucial values? The FRC, the RV, and therefore the TLC, there must be another way.
There is. It requires an indirect measurement method. The most common one described in our source is the helium dilution method. This technique is specifically designed to measure the functional residual capacity, FRC. And once you know the FRC, you can easily calculate the RV and TLC using the other values you did get from simple spirometry.
Helium dilution. Okay, walk me through that. How does it work? What's the principle?
The principle is pretty straightforward. It relies on the fact that an inert gas, like helium, which isn't significantly absorbed by the blood in the lungs, will distribute itself evenly throughout any closed space it's introduced into.
Okay, it just mixes uniformly.
Right. So you start with a spirometer circuit that has a known initial volume of gas inside it, and crucially, a known, precisely measured concentration of helium mixed into that gas.
Known volume. Known helium concentration in the spirometer. Check.
Now, the person breathes normally for a bit. Then, at the very end of a normal, quiet expiration, that precise moment when the volume of air remaining in their lungs is equal to their functional residual capacity, FRC, they are immediately switched into breathing from this closed spirometer circuit.
So the air already in their lungs, the FRC, now gets connected to the spirometer air.
Exactly. The helium, which was only in the spirometer to begin with, now starts to mix with the gas that was already present in the person's lungs, the FRC volume. The helium spreads out and distributes itself evenly throughout the entire combined volume, the spirometer volume plus the unknown FRC volume.
So the helium gets diluted because it's spreading into a bigger total space.
That's the key. Since the total amount of helium in the closed system hasn't changed, it just moved around and mixed, but the volume it occupies has increased by adding the FRC, the concentration of helium must decrease.
And by measuring how much the helium concentration drops, you can figure out how big that unknown lung volume, FRC, was.
Precisely. After the person breathes in this closed circuit for a short time, long enough for the helium to mix completely and the concentration to stabilize, you measure the new, lower, final concentration of helium in the spirometer. Since you know the initial spirometer volume, VSPIR, the initial helium concentration, CRE, and the final helium concentration, CFEG, you can use a simple dilution equation to calculate the unknown volume the helium mixed into, which is the FRC. The source provides the formula. FRC equals initial HECONC, final HECONC, 1 initial spirometer volume, or using the symbols, FRC equals CARE, CFEG, 1 SFIR.
That is quite clever. The ratio of the starting concentration to the ending concentration tells you exactly how much the original volume of helium got spread out. And that dilution factor is directly related to the total volume it spread into, so you can just mathematically solve for the FRC.
It's a really elegant application of basic physics to measure a lung volume that's otherwise inaccessible to direct measurement. And once you've determined the FRC using this helium dilution method, calculating the residual volume, RV, and the total lung capacity, TLC, becomes straightforward. You just combine the FRC value with the volumes you can measure directly using the standard spirometry.
How do you get RV and TLC from FRC then?
Easy. Remember, functional residual capacity is defined as expiratory reserve volume plus residual volume, FRC, ERV plus RV. You measure the ERV using simple spirometry. So you can calculate the residual volume simply by subtracting ELV from the FRC you just found. RV equals FRC ERV.
Okay.
Similarly, total lung capacity is defined as functional residual capacity plus inspiratory capacity, TLC, FRC plus IC. You've calculated FRC, and you measured the IC with spirometry. Remember, IC, VT plus IRV. So you calculate TLC by adding them together. TLC, FRC plus IC.
Okay, so simple spirometry gives you VT, IRV, ERV, IC, and VC. But you need a trick like helium dilution to get the FRC. And once you have FRC, that unlocks the RV, FRC, ERV, and the TLC, FRC plus IC. It ties the direct measurements together with an indirect one to paint the full picture of lung volumes. That seems like a really key concept to grasp for mastering this stuff.
It absolutely is. Understanding not just the definitions, but also the measurement techniques for these different volumes and capacities is fundamental to how lung function is assessed in both health and disease.
All right. We've talked about how much air can be in the lungs or how much can be moved in a single maximal effort. But ultimately, for everyday life, what really matters is how much air is actually moving in and out per minute. And maybe even more importantly, how much of that air moving per minute is truly useful for gas exchange.
Exactly. This brings us to the crucial difference between minute respiratory volume and alveolar ventilation. Minute respiratory volume is the simpler concept. It's just the total amount of new air moved into the respiratory passages each minute.
And that's just a simple multiplication.
Very simple. It's the respiratory rate, how many breaths you take per minute, multiplied by the tidal volume, how much air you move with each breath. Minute respiratory volume, rate tidal volume.
That's the formula. So for our normal, healthy adult man at rest, the tidal volume, VT, is about 500 milliliters, and the respiratory rate is around 12 breaths per minute. Therefore, the average minute respiratory volume is 12 breaths per 500 milliliter breath equals 6,000 milliliter, or 6 liters per minute. That's the total airflow rate into the system.
And what are the extremes? How low or high can that go?
Our source notes that a person can actually survive, at least for short periods, with a minute respiratory volume as low as 1.5 Lmin, achieved by breathing very slowly, maybe only 2 to 4 breaths per minute. At the other extreme, during maximal exercise, the respiratory rate can shoot up to 40 or 50 breaths per minute, and the tidal volume can become as large as the entire vital capacity, around 4,600 milliliters in our example. If you multiply those maximums, theoretically, you could get a minute respiratory volume over 200 millimin.
Wow, over 200 liters a minute. Huge potential airflow. However, the source clarifies that most people can only sustain about half to two-thirds of their absolute maximum minute volume for more than a minute or so. Still, the range is enormous, from 1.5 Lmin up to maybe 100, 120 Lmin sustainably.
Okay, so 6 liters per minute normally at rest, but capable of moving massive amounts when needed. But you mentioned not all of that air is actually useful.
That's the critical distinction we need to make for understanding gas exchange. The ultimate purpose, the whole point of pulmonary ventilation, is to continually renew the air down in the gas exchange areas of the lungs. These are the specific zones where the inspired air comes into intimate contact with the pulmonary blood. We're talking about the alveoli themselves, but also the alveolar sacs, the alveolar ducts, and the respiratory bronchioles leading directly into them. The rate at which new fresh air reaches these specific gas exchange areas is called alveolar ventilation, VA.
So alveolar ventilation is the effective ventilation for gas exchange. What air doesn't reach those areas then? Where does the rest of that minute respiratory volume go?
That air occupies the dead space. Anatomical dead space, specifically. This is the volume of air that fills all the conducting respiratory passages where no significant gas exchange occurs. Think about the path air takes. Nose, pharynx, larynx, trachea, the main bronchi, and all the branching bronchioles down to the point where alveoli start appearing, the terminal bronchioles. Air filling this conducting tree is called dead space air because it doesn't participate in exchanging O2 and CO2 with the blood. It's just along for the ride down and back up.
And when you exhale, this wasted dead space air from the tubes is the first bit of air that comes out?
Exactly right. The air that was just sitting in your trachea and bronchi from the previous breath gets pushed out first, before the air from your alveoli that actually underwent gas exchange. This makes the process of clearing waste gases like CO2 slightly less efficient than if 100% of the inhaled air went directly to functioning alveoli.
How do they measure the volume of this anatomical dead space?
There's a clever experimental method described in the source, using a rapid nitrogen meter. It's illustrated in figure 38-7. Here's how it works. The subject first takes a single deep breath of 100% pure oxygen.
Okay, filling the lungs with oxygen.
Right. This pure O2 completely fills all the dead space passages. It also goes down and mixes with the air already in the alveoli air, which, under normal conditions, breathing room air, contains a high concentration of nitrogen from all the previous breaths.
So dead space has pure O2, alveoli have a mix of O2 and N2.
Correct. Then the subject immediately exhales forcefully and completely through a special meter that can continuously measure the concentration of nitrogen N2 in the expired air second by second.
What does that nitrogen meter show as they breathe out?
The recording is the key. The very first portion of air expired comes directly from the dead space, trachea, bronchi, etc., which was just filled with 100% oxygen. So this initial expired air contains 0% nitrogen.
Makes sense.
As the expiration continues, air from the alveoli, which is rich in nitrogen, starts to be expelled and begins to mix with the last bits of the oxygen-filled dead space air still being cleared. So the recorded nitrogen concentration rises rapidly as this mixed air comes out. Finally, after all the dead space volume has been washed out, only pure alveolar air is being expired. At this point, the nitrogen concentration plateaus at the stable, high level that reflects the N2 concentration in the alveoli.
And you can calculate the dead space volume from that graph.
Yes. The volume of expired air during which the nitrogen concentration was essentially zero corresponds to the volume of the anatomical dead space. Our source describes a way to calculate it using the areas on the recorded graph. The dead space volume, VD, equals area representing the initial pure dead space air, divided by total area representing all expired air, all multiplied by the total expired volume, VD. Using the example numbers from the text, a gray area of 30 square centimeters represented the 0% N2 dead space air, and a pink area of 70 square centimeters representing the nitrogen-containing alveolar air, with a total expired tidal volume of 500 milliliters. The calculation works out as VD equals 30, 70 plus 30, 500 milliliter, 150 milliliters. 150 milliliters, so in a typical 500 milliliter tidal breath, about 150 milliliters of that inhaled air just fills the conducting pipes and never gets down to the gas exchange level. That's the anatomical dead space. Yes, that 150 milliliters is the typical volume of the conducting airways, the anatomical dead space, in a healthy young adult man. It tends to increase slightly with age. However, the source introduces another absolutely vital distinction here, the concept of physiological dead space.
Physiological dead space, how is that different from anatomical?
The physiological dead space includes all the volume within the respiratory system that does not participate in gas exchange. It is defined as the anatomical dead space, plus any alveolar dead space.
Alveolar dead space, what's that?
Alveolar dead space refers to the volume of air that might reach alveoli, but those specific alveoli are non-functional or only partially functional when it comes to gas exchange. This typically happens if the alveolus is ventilated, gets air, but has inadequate or even absent blood flow through the surrounding pulmonary capillaries.
So air gets into the sac, but there's no blood flowing past it to pick up O2 or drop off CO2.
Exactly. If air gets there, but there's no perfusion, blood flow, for exchange to happen, that volume of air within that alveolus is also effectively wasted in terms of contributing to gas exchange. It becomes part of the physiological dead space.
So anatomical dead space is the pipes. Physiological dead space is the pipes plus any alveoli that get air but have no blood flow.
You've got it. Now, in healthy lungs, where blood flow perfusion is generally well matched to ventilation in almost all alveoli, the anatomical and physiological dead spaces are nearly identical. Maybe physiological is just a tiny bit larger, 5-10 milliliters perhaps. Basically, all alveoli that get air also get blood flow.
But in lung disease, I bet that changes.
Dramatically. In many pulmonary diseases, things like pulmonary embolism, blood clots in lung arteries, emphysema, destruction of alveolar walls and capillaries, or conditions causing low cardiac output blood flow to certain parts of the lungs can be severely reduced or completely blocked, even if air can still get into the alveoli in those regions. In such cases, the alveolar dead space can become very large. Consequently, the physiological dead space can become much, much larger than the anatomical dead space sometimes, as the source notes, up to 10 times larger, potentially reaching 1 to 2 liters of wasted ventilation volume.
Wow, 1 to 2 liters of dead space. That's huge.
It is. Our source points out this concept is critical for understanding gas exchange problems in disease and is covered more later. But the distinction is fundamental for understanding effective ventilation right now.
That's a really key insight for mastery, I think. A person might be moving a normal total volume of air, minute ventilation, and have a normal anatomical dead space, the airways. But if they have a large physiological dead space because of poor blood flow matching, a huge fraction of every breath they take is completely useless for gas exchange. It totally changes how you interpret their breathing pattern and effort.
Okay, now with this concept of physiological dead space in hand, how do you calculate the truly useful air movement, the alveolar ventilation rate?
Right, the alveolar ventilation rate, of VA, is the total volume of new, fresh air that actually reaches the functional alveoli and other gas exchange areas each minute. It's the rate of effective ventilation. The formula is alveolar ventilation rate, respiratory frequency, breaths min, tidal volume, physiological dead space volume.
VA freak, VTVD, where that VD specifically means the physiological dead space. Precisely. So let's use our normal example values again. 12 breaths per minute, 500 milliliter tidal volume, 150 milliliter physiological dead space, 12 breaths min, 350 milliliter effective volume per breath, 4,200 milliliters per minute, or 4.2 liters per minute.
Okay, so while the total air moved into the passages, my net respiratory volume was 6 lin, the truly useful air actually reaching the exchange surfaces, alveolar ventilation, is only 4.2 lemon. That 1.8 lemon difference is the ventilation that's just washing in and out of the dead space with each breath.
Correct. And it's this alveolar ventilation rate, VA, that is the major factor determining the actual concentrations of oxygen and carbon dioxide down in the alveoli. And those concentrations, in turn, are what drive the diffusion of gases across into the blood. So understanding and being able to calculate alveolar ventilation is absolutely central to understanding pulmonary gas exchange.
Which raises an interesting question. Why does the body even bother having this anatomical dead space, all these conducting airways when no exchange happens? It seems inherently inefficient to move 150 milliliters of air back and forth with every breath just to fill the pipes.
That is a very pertinent question. And part of the answer lies in the necessary structure required to efficiently conduct air from the single trachea down to the literally hundreds of millions of tiny alveoli, providing a huge surface area. But the structure of those air passages themselves is critical. They need to stay open, they contribute to resistance, and they can even be actively controlled. Which leads us right into our next topic. The air passageways themselves, their structure, how they affect airflow resistance, and how their diameter is regulated.
Okay, so air has to travel through this complex branching network of tubes to get down to the alveoli. What are these passages like structurally? And what keeps them from collapsing, especially when pressures inside and outside are changing with each breath?
Right. The respiratory passages form this intricate branching tree, specifically designed for conducting and distributing air efficiently, but also incorporating features to keep them open and protected from collapse. Figure 38-8 in the source gives a nice overview of this pathway, from the large trachea down to the microscopic air sacs.
Starting at the top with the trachea, the windpipe.
The trachea is the largest airway. It's kept from collapsing, kept rigid, by multiple C-shaped rings of cartilage. These rings extend around about 5/6 of its circumference. The back part is soft muscle, allowing the esophagus behind it some room. But these rings provide significant structural support.
Okay, cartilage keeps the trachea open. What about as it branches into the bronchi?
As the trachea splits into the two main bronchi, and as they branch further, you still find cartilage, but it's in the form of less extensive curved plates rather than complete rings. These plates maintain a reasonable amount of rigidity in the larger bronchi, preventing collapse. But they also allow for some flexibility in motion, as the lungs themselves expand and contract within the chest.
And further down the tree, as the airways get smaller and smaller.
As the passages continue to branch into smaller bronchi, and then into the even smaller bronchioles, those cartilage plates become progressively less extensive. Eventually, they disappear completely in the bronchioles, which are typically defined as airways having diameters less than about 1.5 millimeters.
So the bronchioles are really small airways right before you get to the respiratory bronchioles and alveoli. They don't have any cartilage supporting them. What keeps them from collapsing, especially during expiration, when the pressure outside them in the lung tissue might be trying to squeeze them shut?
This is a really key point from the source material, fundamental to understanding airway function and disease. The bronchioles lack rigid cartilage walls. They are kept expanded, kept open, mainly by the same transpulmonary pressure that expands the alveoli around them.
How does that work?
Think of it this way. The bronchioles are embedded within the elastic lung tissue, like tunnels through a sponge. As the chest wall pulls outwards during inspiration, increasing the transpulmonary pressure, the pressure difference across the lung tissue, this stretches the surrounding alveolar walls open. This outward pull, or radial traction, from the surrounding stretched lung tissue is also exerted on the walls of the bronchioles embedded within that tissue. This pull holds the bronchioles open. As the alveoli enlarge, the bronchioles running through them are also pulled open, although maybe not quite to the same degree.
So the bronchioles basically rely on the surrounding lung sponge being stretched open by the overall lung inflation, driven by that negative pleural pressure, to keep them patent. That makes them inherently vulnerable then if that outward pull is weak, like if the lung tissue loses its elasticity.
Absolutely. If that radial traction from the surrounding lung tissue is reduced, for example, in emphysema where the elastic alveolar walls are destroyed, or if the transpulmonary pressure decreases significantly, the bronchioles are much more likely to collapse, especially during expiration when pressures are trying to compress them anyway. This bronchiolar collapse is a major factor in the airflow limitations seen in diseases like COPD.
Okay. What about the walls of these passages in the areas where there isn't cartilage, like the back of the trachea, or especially in those bronchioles?
In the parts of the trachea and larger bronchi that aren't supported by cartilage, like the posterior wall, the walls contain smooth muscle. As you move down into the smaller bronchioles, the walls become almost entirely composed of smooth muscle, with very little other structural support besides the surrounding tissue. The only exception is the very terminal sections, the respiratory bronchioles, where gas exchange begins. They have minimal smooth muscle and are mainly just thin epithelium and fibrous tissue.
And the smooth muscle in the bronchi, and especially the bronchioles, that's clinically significant.
Critically significant. Our source states that many obstructive lung diseases like asthma and also chronic bronchitis involve significant narrowing of these smaller bronchi, and particularly the mucking bronchioles. This narrowing, this obstruction to airflow, is often due to excessive abnormal contraction of this smooth muscle. And frequently, this muscle contraction is combined with swelling edema developing in the airway walls, or excessive mucus accumulating within the airway lumen, further blocking the passage.
Okay, so the structure transitions. Rigid cartilage higher up, then mainly smooth muscle lower down, with the smallest airways critically dependent on the outward pull from the surrounding lung tissue to stay open. How does airflow resistance fit into this picture? Where does the air encounter the most difficulty flowing?
Under normal, healthy conditions, airflow through this whole system is actually very easy. Remember, we said only a tiny pressure gradient, less than 1 centimeter H2O difference between the alveoli and the atmosphere, is needed to move the air required for quiet breathing. Now, what might be surprising is where the greatest resistance to this airflow normally occurs.
My first guess would be in those tiniest terminal bronchioles, just because they're individually so narrow.
That's a very common intuition, and it makes sense thinking about a single tube. But according to our source, the greatest amount of airway resistance in a healthy lung actually occurs not in the multitude of tiny terminal bronchioles, but in some of the larger bronchioles and bronchipores located closer to the trachea.
Really? Why is that the case? The bigger pipes have more resistance.
It comes down to the network architecture and the total cross-sectional area available for flow at each level. While each individual terminal bronchiole is indeed very small and narrow, there is an absolutely enormous number of them branching out in parallel. The source estimates around 65,000. The total airflow gets divided amongst these countless tiny parallel pathways.
Like lanes on a highway.
Exactly. Each terminal bronchiole only has to handle a minuscule fraction of the total flow. In contrast, there are far, far fewer of the larger bronchi and bronchioles higher up in the tree, closer to the trachea. Even though each one of these is individually much wider, their limited number means that collectively, the total cross-sectional area available for airflow at that level is actually smaller than the combined cross-sectional area of all those 65,000 terminal bronchioles added together. Therefore, these larger passages, despite being wider individually, end up offering greater resistance to the bulk flow of air moving through them compared to the vast network of tiny parallel passages further down.
Okay, that makes sense now. It's about the cumulative bottleneck effect of fewer larger pipes versus the massive combined open area of countless tiny parallel pipes, like traffic merging from many small roads onto a slightly wider but single highway.
That's a good analogy. However, the source is very careful to emphasize that in disease states, the situation can flip dramatically. In conditions like asthma or bronchitis, where the walls of the smaller bronchioles become narrowed due to muscle spasm, inflammation, swelling, and mucus, then these smaller airways absolutely do become the primary sites of increased resistance. Because they are small in diameter to begin with, even a slight amount of further narrowing has a much greater proportional effect on resistance. Resistance is inversely proportional to the radius of the fourth power, compared to the same degree of narrowing in a larger airway.
Right, so normally resistance is highest higher up, but disease often shifts the major problem down to those smaller, more easily occluded bronchioles. which highlights, again, the clinical relevance of that smooth muscle we talked about controlling their diameter. How is the contraction and relaxation of this bronchiolar smooth muscle controlled by the body?
Right. The diameter of these muscular airways is actively regulated by both nervous signals and various local chemical factors. Direct control via sympathetic nerve fibers that actually run to the lung tissue itself is thought to be relatively weak in humans. However, the bronchial tree is highly responsive to circulating hormones called catecholamines, particularly epinephrine and adrenaline, and, to a lesser extent, norepinephrine. These are released into the bloodstream from the adrenal medulla, typically as part of the sympathetic nervous system's broader fight-or-flight response.
So adrenaline affects breathing airways. How?
Absolutely. Epinephrine strongly stimulates specific receptors on the bronchial smooth muscle called beta-adrenergic receptors. Activation of these receptors causes the smooth muscle to relax, resulting in significant dilation, widening of the entire bronchial tree. This makes breathing easier. That's why inhaled beta-agonist drugs, which mimic epinephrine's effect on these receptors, are the cornerstone of treatment for asthma attacks. They open up the airways.
Okay, sympathetic stimulation via circulating adrenaline causes dilation. What about the other side of the autonomic nervous system, the parasympathetic?
The parasympathetic nervous system also plays a role. Nerve fibers originating from the vagus nerves do penetrate deep into the lung tissue, the parenchyma, and directly innervate the bronchiolar smooth muscle. When these vagal nerve fibers are activated, they release the neurotransmitter acetylcholine. Acetylcholine acts on different receptors, muscarinic receptors, on the smooth muscle, and its effect is the opposite. It causes mild to moderate constriction, narrowing of the bronchioles.
And that has clinical implications too.
Significant ones, yes. If a person already has some baseline airway narrowing due to a disease like asthma, any increase in parasympathetic activity can worsen their symptoms by causing further bronchoconstriction. This is why drugs that block the action of acetylcholine, called anticholinergics, like atropine or the commonly used inhaled drug epitropium, can be helpful clinically. They relax the airways by blocking this parasympathetic constrictor tone, providing relief from obstruction.
Can these parasympathetic nerves, the ones causing constriction, be activated reflexively, like triggered by something happening in the lungs?
Yes, absolutely. The parasympathetic pathways leading to bronchoconstriction can be triggered by reflexes that originate right there in the airways or lungs. These reflexes can be set off by irritation of the airway epithelial, lining the sensitive inner surface by things like inhaled noxious gases, dust particles, cigarette smoke, or even inflammation during a bronchial infection. Interestingly, the source also mentions that a bronchoconstrictor reflex can be triggered by tiny blood clots, microemboli, lodging in and blocking small pulmonary arteries. The presumed purpose here might be to reflexively divert airflow away from areas of the lung that are no longer receiving adequate blood flow.
Wow. So the airways can constrict automatically in response to inhaled irritants, infections, or even problems with blood flow, all mediated by these nervous reflexes. Are there also substances produced locally right there in the lung tissues that can affect airway diameter without involving nerves?
Yes, definitely. Several potent chemical mediators can be released within the lung tissues themselves, and these can cause significant local changes in airway diameter. Two of the most important examples highlighted in our source are histamine and a group of compounds collectively called slow reactive substance of anaphylaxis, SRSA, which include leukotrienes.
Histamine, that's the classic allergy chemical, right?
Correct. Both histamine and SRSA are released in large amounts from specialized immune cells called mast cells, which are abundant in lung tissues. This release is particularly triggered during allergic reactions, for instance, when someone inhales an allergen they're sensitive to, like pollen or dust mite particles. Both histamine and SRSA cause powerful contraction of bronchial smooth muscle, leading to significant bronchoconstriction and airway narrowing. Our source specifically notes that SRSA, leukotrienes, is considered a major player in causing the severe, prolonged airway obstruction seen during episodes of allergic asthma.
And can other inhaled irritants act directly on the airways too, besides triggering reflexes?
Yes. Beyond just setting off those nervous reflexes, many inhaled irritants, things like smoke, dust, sulfur dioxide, SO2 from pollution, and various acidic components of smog, can also have direct irritant effects on the airway tissues themselves. This can initiate local inflammatory responses or direct contractile responses in the smooth muscle, contributing further to the airway obstruction, independent of the main nerve pathways.
Okay, so the airways are this incredibly dynamic system. You've got the rigid tubes higher up, then the muscular sections in the middle, and then the smallest airways that rely critically on the surrounding lung tissue's outward pull to stay open. And the diameter of those muscular airways is constantly being fine-tuned by a whole host of factors. Circulating hormones like adrenaline causing dilation, direct nerve signals from the parasympathetic system causing constriction, which can be reflexively triggered by all sorts of things, and potent local chemical mediators like histamine and SRSA released during inflammation or allergic reactions causing constriction, plus direct irritation effects. It's amazing how many layers of control there are, and also, unfortunately, how many ways things can go wrong and lead to airway narrowing in diseases like asthma or COPD.
It truly is a complex interplay. Structure, mechanics, nervous control, local chemical control, all interacting for precise regulation, but also creating multiple points of vulnerability when things get disrupted.
Now, beyond just getting air down to the alveoli and controlling the pipe size, the respiratory passages have another absolutely essential job. Keeping themselves clean. We're breathing in air that's full of dust, microbes, pollution. How does the body prevent all that gunk from reaching and damaging the delicate gas exchange surfaces deep inside?
This is a critical protective function, absolutely vital. And the respiratory system has evolved multiple really elegant mechanisms to handle this continuous challenge. First off, almost all the surfaces of the respiratory passages, right from the nasal cavity down to the very end of the conducting zone, the terminal bronchioles, are lined with a thin, sticky layer of mucus.
Where does this mucus come from?
It's secreted primarily by two sources. Individual mucus goblet cells that are scattered throughout the surface epithelial lining, and also small submucosal glands located just beneath the surface epithelium in larger airways.
And what's the mucus for? Just keeping things moist?
Keeping the surfaces moist is important, yes. It prevents drying and cracking of the delicate tissues and is needed for proper cell function. But crucially, the mucus acts as a sticky trap. Small particles from the inspired air that make it past the nose and come into contact with this mucus layer get stuck fast, physically preventing them from penetrating any deeper into the lungs.
Okay, so the mucus traps the incoming particles, but then how does that trapped stuff get removed? It can't just build up forever.
Right. That's where the second part of the system comes in, the cilia. This mechanism is often called the ciliary escalator, or the mucociliary escalator, and it's ingenious. The entire surface of the respiratory passages, again from the nose down to the end of the terminal bronchioles, is covered with ciliated epithelium. Each one of these epithelial cells lining the airways is equipped with about 200 tiny hair-like projections sticking out from its surface. These are the cilia.
And they're constantly moving, beating.
Yes, they beat continually and very rapidly, at a frequency of something like 10 to 20 times per second. But the absolute key is the direction of their beating. The effective power stroke of the cilia, the direction they push in, is consistently oriented towards the pharynx, the throat. This means that the cilia in the lungs and lower airways beat upwards, while the cilia lining the nasal passages beat downwards towards the throat.
Ah, so they're all pushing the mucus layer in one direction towards the exit, like a microscopic conveyor belt.
Exactly like a conveyor belt. This coordinated directional beating causes the entire sheet of mucus, along with all the particles, microbes, and other debris trapped within it, to be moved slowly but continuously upwards from the lungs or downwards from the nose toward the pharynx. Our source estimates this flow rate is only a few millimeters per minute, but it's constant.
And what happens when it reaches the pharynx, the throat?
Once the mucus and its entrapped load reach the pharynx, it's then typically, and usually unconsciously, swallowed. It goes down the esophagus into the stomach, where the acid destroys most microbes and the debris is eliminated via the digestive tract. Occasionally, if there's a large amount, it might trigger a conscious need to cough it up and expectorate, spit it out.
That's a brilliant, continuous, low-energy, self-cleaning system. It's just always working away in the background, sweeping the airways clean. But what if something bigger gets in? Or there's a sudden major irritation? Is there a backup plan?
Yes, absolutely. For more substantial clearance needs or acute threats, the body employs powerful explosive reflexes. The most prominent of these is the cough reflex.
Okay, the cough. What triggers it? Where are the sensors?
The sensory nerve endings that trigger the cough reflex are most abundant and sensitive in the lining of the lower airways, particularly the bronchi and the trachea, and especially at the carina, which is that ridge where the trachea splits into the two main bronchi. These areas are exquisitely sensitive to even light touch or the presence of any foreign matter. However, the source also notes that inhaled corrosive chemical irritants like sulfur dioxide or chlorine gas can also trigger the reflex, sometimes even from receptors located way down in the terminal bronchioles or alveoli.
And how does the reflex actually work? What's the sequence of events?
When these sensory receptors are stimulated, afferent nerve impulses shoot up to the brainstem, primarily via the vagus nerves, activating a specialized cough center in the medulla. This center then orchestrates a very rapid, automatic sequence of motor commands. First, you take a quick, deep inspiration, often drawing in as much as 2.5 liters of extra air.
A big gulp of air in first.
To get a large volume of air behind the potential obstruction, to provide the force for expulsion. Next, the epiglottis, the flap that covers the top of the larynx, slams shut, and the vocal cords close tightly over the glottis. This effectively traps that large volume of inspired air within the lungs.
Sealing the exit.
Exactly. Then comes the forceful part. There is a sudden, strong, coordinated contraction of the abdominal muscles, pushing the diaphragm sharply upwards, and the internal intercostal and other expiratory muscles. This contraction exerts immense pressure on the trapped air within the lungs. The pressure inside the lungs can rise incredibly rapidly, sometimes reaching as high as 100 millimeters of mercury, millimeter Hg, or even more.
Building up massive pressure behind the closed door.
Precisely. Finally, the vocal cords in the epiglottis are suddenly thrown open widely. The air that was trapped under this extremely high pressure is instantly expelled outwards with tremendous force and velocity. Our source states the expelled air can reach speeds ranging from 75 to 100 miles per hour.
Whoa, a physiological air blast. That's incredible speed.
It is. And there's another important part of the mechanism. During that phase of forceful compression just before the opening, the high pressure inside the chest actually causes the non-cartilaginous parts of the trachea and larger bronchi to collapse inwards slightly. This temporarily turns the normally round airways into narrowed slits.
Okay, why does that happen?
It seems counterintuitive, but it's crucial. The exploding air is then forced at that incredibly high velocity through these narrowed slits. This high-speed jet of air, shearing through the collapsed airways, is extremely effective at dislodging and propelling any foreign matter or excess mucus outwards, clearing the passages.
Ah, that makes perfect sense. The high-speed flow through a deliberately narrowed opening acts like a power washer, scouring the walls and blasting debris out. What about the other big reflex, the sneeze? How is it different?
The sneeze reflex is very similar in its overall mechanism to the cough, deep inspiration, closure, pressure buildup, forceful expulsion. But the trigger is different, and the exit route is different. A sneeze is triggered by irritation specifically in the nasal passages, not the lower airways. Efferent nerve impulses travel via the fifth cranial nerve, the trigeminal nerve, to the medulla. The motor sequence is similar, but during a sneeze, the uvula, that little fleshy pendulum hanging at the back of your throat, is actively depressed.
What does depressing the uvula do?
It partially closes off the opening from the pharynx down into the mouth and larynx. This action directs a large portion of the forcefully expelled air rapidly through the nose instead of just out the mouth. So a sneeze specifically serves to clear the nasal passages of irritants or foreign matter.
Okay, so the body has this amazing multi-layered defense, the continuous, quiet cleaning crew of the mucus and cilia working 24/7, and then these powerful, explosive backup reflexes coughing, targeted at clearing the lower airways, sneezing targeted at clearing the nose, ready to deploy when a bigger, more acute problem arises. It really is a robust system to keep those vital air passages open and clean.
It truly is. A testament to how critical maintaining airway patency and cleanliness is for survival, reflected in these elegant and powerful biological mechanisms.
Now, speaking of the nose and clearing particles, the nose itself does a huge amount of the initial filtering and also conditioning of the air we breathe right at the very start of the journey, doesn't it?
It absolutely does. The nasal passages perform several critical air conditioning functions before the inspired air even gets close to the lower, more sensitive parts of the respiratory system, like the trachea and lungs. Our source highlights 3 essential functions performed by the nose, and you can visualize the structures involved in figure 38-8.
Okay, what are those 3 main nasal functions?
First, the inspired air is warmed. As the air flows turbulently over the extensive, highly vascularized surfaces of the nasal conchae, also called turbinates, those folded bony shelves inside the nose, and the nasal septum, the dividing wall, heat is rapidly transferred from the blood-rich tissues to the air. The total surface area available for this warming inside the nose is surprisingly large, estimated at about 160 square centimeters.
A big radiator surface crammed into a small space to warm the air quickly. Clever.
Exactly. Second, the air is almost completely humidified. As it passes through the nasal passages, it flows over the wet mucous membranes lining these surfaces, and water evaporates from the mucus into the air, saturating it with water vapor. This happens even before the air goes down the pharynx and trachea.
So it warms it and wets it, and the third function is filtering, you said.
Yes, and it's very effective filtering. Particles are removed from the airstream as it passes through the nose. These three functions together, warming, humidifying, and filtering, are often collectively referred to as the air conditioning function of the upper respiratory passages.
What's the net result of all this nasal air conditioning by the time the air actually reaches the trachea, heading down to the lungs?
Ordinarily, by the time inspired air gets to the trachea, its temperature is typically within just 1 degree Fahrenheit of the core body temperature, and its humidity is within just 2% to 3% of being fully saturated with water vapor at that temperature. This warm, moist air is ideal for the delicate tissues of the lower airways and alveoli. It prevents them from drying out or getting chilled.
And the source underscores how important this is by talking about what happens if you bypass the nose.
Exactly. The source mentions the consequences of breathing directly through a tracheostomy tube inserted into the trachea. Without the nose's highly efficient warming and humidifying action, the relatively cold, dry atmospheric air goes straight into the lower airways. This has significant cooling and especially drying effects on the bronchial and alveolar surfaces. This drying can lead to serious problems, including the formation of thick, hard crusts of secretions, inflammation of the tissues, and a much higher susceptibility to serious pulmonary infections.
Wow, that clearly demonstrates just how critical that seemingly simple nasal air conditioning function is for protecting the lungs lower down. How does the filtration mechanism in the nose work? Is it just the hairs at the entrance?
The hairs right inside the nostrils do trap very large particles, yes. But the much more important mechanism for removing smaller, inhaled particles within the nasal passages is called turbulent precipitation.
Turbulent precipitation. How does that work? Sounds like making it rain particles.
Sort of. As the inspired air flows through the complex, winding, narrow passageways of the nasal cavity, it's forced to make many sharp turns and encounters numerous obstructions. These include the projecting conchae, turbinates, the nasal septum, and eventually the back wall of the pharynx. Every time the stream of air hits one of these surfaces, it's forced to change direction abruptly. This creates turbulence.
Okay, lots of swirling air.
Now, the particles suspended in that air dust, pollen, microbes, have significantly more mass and therefore more momentum inertia than the air molecules themselves. Because of this inertia, they cannot change direction as rapidly as the airflow when it hits an obstruction.
So they tend to just keep going straight, like a car trying to take a sharp turn too fast.
Exactly. They tend to continue traveling in their original direction, momentum carrying them forward until they collide with or precipitate onto the surfaces of those obstructions, the conchae, the septum, the pharyngeal wall. And remember, these surfaces are coated with that sticky layer of mucus, so the particles get stuck firmly in the mucus upon impact, effectively removed from the airstream. Then, as we discussed, the cilia sweep this mucus particle mixture backwards towards the pharynx to be swallowed.
It's an elegant, natural inertial impaction filter using turbulence and particle momentum. How effective is this nasal filtration for particles of different sizes?
It's highly effective, especially for relatively larger airborne particles. Our source states that due to this turbulent precipitation mechanism, almost no particles larger than 6 micrometers in diameter usually manage to enter the lungs through the nose. For perspective, 6 micrometers is smaller than the diameter of a red blood cell.
So the nose catches most of the significant larger debris right away. What happens to smaller particles that do make it past the nose and into the lower airways?
Particles in the intermediate size range, roughly between 1 and 5 micrometers, often manage to follow the airflow down into the smaller bronchioles. However, many of these tend to settle out in these smaller airways before reaching the alveoli, primarily due to gravitational precipitation. As the airflow velocity slows down dramatically in the progressively smaller and vastly more numerous airways, gravity has more time to act on these mid-sized particles, pulling them downwards out of the suspended airflow and onto the airway walls. The source gives coal miner's lung, coworkers pneumoconiosis, as an example of a disease where inhaled coal dust particles, often in this 1 to 5 micrometer size range, settle predominantly in the terminal bronchioles, causing localized inflammation and fibrosis there.
Okay. And what about even smaller particles, like less than 1 micrometer? They're really fine stuff.
Particles smaller than 1 micrometer in diameter are generally not effectively filtered out by either turbulent precipitation in the nose or gravitational settling in the airways before they reach the alveoli. Many of them remain suspended in the airflow all the way down. Some of these very small particles, particularly those between about 0.5 and 1 micrometer, can eventually diffuse from the air to the alveolar walls and adhere to the fluid lining due to random Brownian motion. However, a large fraction of particles that are less than 0.5 micrometers in diameter, a category that includes many components of cigarette smoke, which the source notes are typically around zero point 0.3 micrometers, are not significantly precipitated in the conducting passages before reaching the alveoli.
So the very smallest stuff gets deep access easily.
It does. The source estimates that perhaps up to 1/3 of these very fine 0.5 micrometer inhaled particles do eventually precipitate out in the alveoli by diffusion, given enough time. But the rest, often the majority, remain suspended in the alveolar air and are simply expelled back out during the next expiration.
So the filtration system is great for bigger stuff, okay, for medium stuff, but pretty poor for the really tiny particles like smoke, which can penetrate right to the deepest, most delicate gas exchange surfaces. What happens to the particles that do make it all the way down to the alveoli and get deposited there? How does the body deal with them?
Particles that land in the alveoli, which lack the protective mucus layer and cilia of the airways, are primarily dealt with by alveolar macrophages. These are specialized immune cells, phagocytes, that constantly patrol the alveolar surfaces. They engulf, eat foreign particles, microbes, and cellular debris that they encounter. Once engulfed, some macrophages might be cleared up the mucociliary escalator if they migrate back to the terminal bronchioles, while others might migrate into the lung tissue interstitium and eventually be carried away via the lung lymphatics.
So there is a cleanup crew even in the alveoli.
There is. However, the source provides a crucial warning here. If an excessive number of particles reaches the alveoli and overwhelms these clearance mechanisms, macrophages and lymphatics, problems arise. The accumulation of indigestible particles and the body's persistent inflammatory reaction to them can lead to the damaging growth of fibrous tissue within the delicate alveolar septa, the walls between alveoli. This fibrosis can thicken the gas exchange barrier, destroy capillaries, reduce lung compliance, and ultimately lead to permanent lung damage and functional debility.
This detailed look at nasal filtration, gravitational settling, and the fate of particles reaching the alveoli. It's another vital piece for mastering this. It really highlights how the respiratory system's defenses are highly effective against certain threats, like larger dust particles, but inherently much less effective against others, like the ultrafine particles in smoke or some industrial pollutants. This explains why certain inhaled substances are so particularly damaging to the deepest parts of the lungs. It's a powerful system, yes, but it has specific vulnerabilities based on particle size.
It absolutely reveals those specific vulnerabilities inherent in the physics of airflow and particle deposition within the system's architecture.
Okay. Now, respiration isn't just about the vital, life-sustaining job of moving air for gas exchange. We also take that airflow and harness it for one of the most complex and uniquely human functions: communication through speech. How does the respiratory system actually contribute to us being able to talk?
Right. Vocalization is indeed a remarkably complex process. And while the lungs and the mechanics of breathing provide the necessary power source, the airflow-producing speech involves much more than just exhaling. Our source breaks down the components of speech beyond just airflow. It mentions specific nervous control centers in the cerebral cortex and the brain's respiratory centers coordinating the airflow, and also the crucial role of the structures of the mouth and nasal cavities in shaping the sound. Mechanically speaking, though, it identifies two primary functions involved in creating speech sounds. First is phonation, which is the actual generation of the basic sound, and this is achieved by the larynx, or voice box. Second is articulation, which is the process of shaping that basic sound into recognizable words and sounds, and this is achieved primarily by the structures of the mouth.
Okay, let's focus first on phonation, how the larynx actually creates the sound.
The larynx, which is shown anatomically in figure 38.9a from our source, is specially adapted to function as a vibrator. The key vibrating elements are the vocal folds, more commonly known as the vocal cords. These aren't really cords, but rather folds of tissue that protrude from the lateral walls of the larynx inwards towards the midline. They partially narrow the opening into the trachea, an opening called the glottis. The tension, the position, and even the shape of these vocal folds are precisely controlled by a complex set of small, specialized muscles within the larynx itself.
How do the vocal cords work differently when you're just breathing quietly versus when you're speaking?
Figure 38.9b illustrates this difference clearly. During normal quiet breathing, the vocal cords are held wide apart. This creates a large opening for air to pass easily into and out of the lungs with minimal resistance. However, during phonation, when you intend to make a sound, the vocal cords are actively moved together, or approximated, towards the center of the glottis, narrowing the opening significantly.
Okay, they close the gap.
Right. Then, when air is forced upwards from the lungs by expiratory effort and passes through this narrowed slit between the closely approximated vocal cords, it causes them to vibrate rapidly back and forth. This vibration of the vocal folds is the fundamental source of the sound energy we perceive as voice.
And how do we control the pitch of our voice, making it higher or lower?
The pitch of the sound produced by the larynx is determined mainly by the degree of stretch or tension applied to the vocal cords. Specific muscles within the larynx can pull on the cartilages the vocal cords are attached to, stretching the cords tighter. Tighter cords vibrate at a higher frequency, producing a higher pitch. Conversely, relaxing the tension on the cords makes them vibrate more slowly, producing a lower pitch. Pitch is also influenced, to some extent, by how tightly the cords are pressed together, adducted, and potentially by the mass or thickness of their vibrating edges.
Can you tell me a bit more about the specific anatomy shown in Figure 38.9a that allows this fine control? What are the key parts?
Sure. Inside each vocal fold, running along its edge, is a strong elastic ligament called the vocal ligament. Anteriorly, at the front, these ligaments attach to the inner surface of the large thyroid cartilage, the prominent cartilage that forms the Adam's apple. Posteriorly, at the back, they attach to small, paired cartilages called the arytenoid cartilages, specifically to projections on them called the vocal processes.
Okay, thyroid cartilage at the front, arytenoids at the back, vocal ligaments stretch between them.
Right. And these cartilages themselves form a framework. The thyroid and arytenoid cartilages articulate, form movable joints, from below with the ring-shaped cricoid cartilage, which sits atop the trachea.
So these cartilages provide the anchor points and the movable levers for the vocal cords. How do the muscles act on them to change the tension and position?
It's quite intricate. Specific small muscles within the larynx act on these cartilages. For example, muscles stretching between the thyroid cartilage and the cricoid cartilage below it can cause the thyroid cartilage to rotate forward. Other muscles acting on the arytenoid cartilages can cause them to rotate posteriorly. Both of these movements effectively pull the arytenoid cartilages further away from the thyroid cartilage, thereby stretching the vocal cords attached between them, increasing their tension and raising the pitch.
And what about loosening the cords to lower the pitch?
That's primarily the job of the thyroarytenoid muscles. These muscles lie just lateral to the vocal ligaments, actually running within the vocal folds themselves. When they contract, they pull the arytenoid cartilages towards the thyroid cartilage, effectively shortening the distance between the attachments. This loosens the vocal cords, decreases their tension, and lowers the pitch. Our source also mentions that small slips of muscle fibers within these thyroarytenoid muscles, right in the substance of the vocal cords, can subtly alter the shape and effective mass of the vibrating cord edges. They can make the edges sharper for producing high-pitched sounds, or more rounded and blunt for producing lower, bass-like tones.
And there are other muscles too, just for positioning.
Yes. Several other small muscles run between the arytenoid and cricoid cartilages. These muscles can rotate the arytenoid cartilages inwards or outwards, bringing the vocal cords together, adduction for phonation, or pulling them apart, abduction for breathing. They can also pull the bases of the arytenoid cartilages together or apart. All these fine movements allow for the precise positioning and approximation of the vocal cords needed for all the different vocal maneuvers, from holding them wide open for breathing, to bringing them gently together for normal voicing, adjusting them for whispering, or closing them tightly for coughing or straining, as shown in Figure 38.9b.
Okay, so phonation itself is this incredibly precise, neurologically controlled process, using airflow from the lungs as the power source to vibrate these carefully tensioned and positioned vocal cords within the larynx, with all this intricate muscular control acting on the laryngeal cartilages. What about the second part of speech you mentioned, articulation, shaping that sound?
Right. Articulation is the process of taking the raw sound vibration produced by the larynx and modifying it into the recognizable vowels and consonants that form understandable words. The shaping is achieved primarily by changing the configuration of the structures of the mouth. The major organs of articulation, as our source notes, are things we're all familiar with using when we talk: the lips, the tongue, and the soft palate, the back, fleshy part of the roof of your mouth. Their complex and rapid movements shape the resonant cavities and obstruct or modify the airflow in specific ways to create the distinct sounds of language.
And resonance plays a role too, amplifying or changing the quality.
Absolutely. After the basic sound is generated in the larynx and initially shaped by articulation in the mouth, it's further modified and amplified by resonators. These are essentially the air-filled cavities through which the sound waves pass before they exit the body. The main resonators include the mouth itself, the nose and the connected nasal sinuses, the pharynx, the throat area above the larynx, and even to some extent, the chest cavity. The size and shape of these cavities selectively amplify certain frequencies present in the laryngeal sound source and dampen others. This resonance is what gives each person's voice its unique quality, or timbre.
And you can hear the effect of these resonators easily.
Definitely. The source uses the familiar example of how your voice quality changes dramatically when you have a severe head cold and your nasal passages are blocked. That nasal voice quality directly demonstrates the significant contribution of the nasal cavities and sinuses as resonators to your normal voice sound. When they're blocked, the resonance changes.
This really shows that breathing, while essential for providing the power, is just the starting point for speech. Speech is this remarkable, complex process layered on top, using incredibly precise muscular control in the larynx to create the fundamental vibration, then actively shaping that sound stream with the lips, tongue, and palate, and finally subtly modifying its quality through the fixed and variable resonant cavities of the head and chest. It's amazing how deeply intertwined the basic respiratory system is with higher nervous function and fine motor control to create something as sophisticated and uniquely human as speech.
It absolutely highlights the incredible adaptability and multi-purpose design of the respiratory system, extending far beyond its primary life-sustaining role in gas exchange.
Wow. Okay, we have really taken a thorough deep dive into the mechanics of pulmonary ventilation today, drawing on all the incredible detail packed into the source material. We started way back with the big picture, looking at respiration's four main components, and firmly established ventilation, the mechanics, as that essential first step.
The foundation, yep.
We explored the intricate muscular actions, the diaphragm's primary role, the ribcage movements, the interplay of external and internal intercostals, the accessory muscles kicking in for forced breathing, showing exactly how the chest volume is changed.
We detailed those two key methods.
Then we dissected the absolutely critical pressure dynamics, that crucial, always negative pleural pressure keeping the lungs tethered, the tiny fluctuating alveolar pressure actually driving the airflow in and out, and the resulting transpulmonary pressure, the true measure of the force inflating the lungs against their own elastic recoil. Getting those pressures straight is key.
Understanding those gradients is fundamental.
We gained, I think, a real mastery of lung compliance, what it means, how it's measured, and the forces behind it, uncovering that surprising dominance of surface tension, which led us to the absolutely critical role of surfactant. Understanding how it works, how dramatically it reduces collapsing forces, and why its absence is so devastating in conditions like infant RDS.
That surfactant story is so important.
We looked at how the thoracic cage itself adds stiffness to the system, modifying the overall compliance. And we analyzed the work of breathing, breaking down the energy cost into compliance work, tissue resistance, and airway resistance, and seeing how dramatically that energy expenditure can increase, especially in disease, becoming a limiting factor itself.
From 3% up to potentially 50 times more energy.
We learned the language of lung volumes and capacities. Tidal volume, reserves, residual volume, FRC, vital capacity, total lung capacity, understanding what each represents, and, importantly, how they're measured using spirometry, plus that clever helium dilution technique needed for the FRC and RV. That was key.
Knowing how we get the numbers.
We carefully differentiated between the total air moved per minute respiratory volume, and the truly useful air actually reaching the gas exchange surfaces. Alveolar ventilation, grasping the concept of dead space anatomical versus physiological and how to calculate effective ventilation was crucial.
Making sure we account for wasted ventilation.
We detailed the structure of the air passageways, from the cartilage-supported trachea down to the muscle-walled, traction-dependent bronchioles. We figured out where airflow resistance is normally highest, surprisingly, not in the tiniest tubes, and explored the complex web of control mechanisms, sympathetic dilation via adrenaline, parasympathetic constriction via acetylcholine and reflexes, plus local chemical mediators like histamine and SRSA that regulate airway diameter.
So many layers of control there.
We uncovered the body's elegant defense systems for keeping those airways clean, the continuous mucus ciliary escalator, backed up by the powerful explosive cough and sneeze reflexes for acute threats.
Essential housekeeping.
And finally, we saw how this whole system of airflow generation is cleverly harnessed for speech, through the intricate mechanics of phonation in the larynx, articulation by the mouth structures, and resonance within the body cavities.
It really pulls it all together.
We've truly seen that what seems like an automatic, simple, often unconscious process, just breathing, is, when you look closely like we have today, an absolute marvel of detailed biomechanics. Precise pressure regulation, intricate muscular control, sophisticated fluid dynamics with surfactant, and elegant protective systems. all working together second by second to maintain life and support other vital functions like communication. Understanding these layers of detail exactly as the source material lays them out, that really is the key to mastering this field at a professional level.
It is. Hopefully, navigating this detailed map of pulmonary ventilation gives you, our listener, a profound new appreciation for every single breath you take and for the incredible intricate system that makes it happen, usually so effortlessly. We've seen that ventilation is far from simple. It's this complex, beautiful dance of muscle, pressure, elasticity, surface tension, and precisely regulated airflow, constantly adapting to our needs.
It truly is fundamental, underpinning everything else the respiratory system achieves, and its normal efficiency is just remarkable when you see all the pieces. And as always, to leave you with a final provocative thought, something to mull over based on our deep dive today. Given the detailed mechanisms we explored for warming, humidifying, and especially filtering air in the upper airways, and understanding how relatively poor the defenses are against those ultrafine particles, 0.5 micrometers, that can penetrate deep into the lungs, what might be the subtle, cumulative, long-term consequences for the delicate, deep lung tissues, the alveoli, the capillaries, if someone lives and works for years in an environment that is continuously dry, cold, and laden with those very fine airborne particles, even if they don't have an obvious diagnosed respiratory disease yet?
A lot to consider there about the chronic challenges our modern environments might pose to this incredibly intricate system. Makes you think.